Okay, so this PCO, there's some plants that have adapted mechanisms to try to diminish how frequently this happens. And, or for some plants, they can cut it out and just stop it for long stretches of time. And so if you live in an area where there's maybe frequent drought or maybe high heat, that might be a good strategy to do, try to get away from doing this. And so that's what we're going to talk about next. We have this idea that some plants, whoops, have figured out a solution to this problem through the process of evolution and natural selection and adaptation. So they have these extra little pathways to try to keep the PCO cycle from not happening or not happening very much at all. And now we're getting into areas of the world where if you think about certain areas of the world that might go through periods of drought or high heat or both, then think about plants in the equatorial region, tropical rainforests, subtropical rainforests, really hot desert areas. You can imagine where these plants could be dealing with consistently hot weather and hot and dry, or yeah, droughty, hot and drought prone areas. And so we're going to talk about what they're doing. And so now we're talking about the plants that are native to these habitats, okay, not the ones that are introduced from somewhere else, but they evolved to adapt to these particular habitats. Okay, so the native plants. And so let's start with the C4. So this is named C4 because the first stable product is a 4-carbon compound. So that's why they named it the C4 cycle. And we're looking at tropical, subtropical, where you do have consistently hot temperatures and sometimes you have periods of drought. When they peer inside these plants and look in their leaves in particular, they see that they have a different leaf anatomy. The cells inside the leaf are arranged a little differently compared to C3 plants. So we're going to zoom in to the part of the leaf that are called bundle sheath cells, and then just the regular like parenchyma mesophyll cells that are around the bundle sheath. We're going to see that in this pathway, this cycle, the plants have engineered a situation to allow for physically different locations of certain things happening. So spatial segregation is how they describe it, of the reactions. Now, don't forget, all plants have a C3 cycle, including C4 plants. They have a C3 cycle. It's just that C4 plants have kind of evolved a little extra cycle and they tacked it on to the C3 cycle. All right, so it's their little flourish on how to adapt to these tropical environments. When they do careful studies of these types of plants, they can see that they handle drought better, high temps better than just straight C3 plants. Water stress, they're just better at dealing with those kinds of stresses compared to just a straight C3 plant. And their photosynthetic rate is better, 2 to 3 times better in some nicely controlled studies. So when they started to look at the C4 cycle and they're like, what's the goal here? What is the plant really trying to do? Basically, one of the huge goals is to concentrate CO2 around rubisco. They're forcing a situation where rubisco sees CO2, 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 and hardly any oxygen. And if you can create that situation, then you've really made sure that the PCO cycle won't run. So just swamp, like I think about rubisco in that bubble bath, you know, like I said the other day, in a big white clawfoot tub, lots of bubbles. That's what the C4 cycle is doing. If you think all those bubbles are carbon dioxide, rubisco is just swimming in carbon dioxide, floating around, having a nice, lovely bubble bath. And so that's what C4 plants are trying to accomplish essentially, so that rubisco doesn't even have a chance to take oxygen into its active site. So some ways of showing the anatomy inside the leaf are drawn here. So the C4 is being shown here in figure B. And the bundle sheath cells are those ring of cells around the vein, okay, around the vascular tissue. Okay, so the gray area in the middle is the xylem and the phloem, okay? And then this ring of cells is called the bundle sheath cells. So we're going to zoom into there. And then all of these lighter green cells that they've drawn, remember, those are just parenchyma cells, all right? They call them mesophyll cells. It's another term for them. I think your book is using that, so that's why I kept it. But mesophyll cells really are, they're just parenchyma cells of the leaf, okay? These are like spongy. Technically, these look like they've drawn the spongy mesophyll, right? So here you can see the spongy mesophyll in the C3 leaf up high. And then here's the palisade. So they're slightly different in their morphology, but that's all just parenchyma cells there. They're just spongy versus palisade. Okay, so we need to think about what's happening in the bundle sheaths and what's happening in the mesophyll cells. They spatially will separate certain things from happening, and it's quite ingenious how they have done this. So here in the lighter green is your mesophyll cell, and then the dark green is your bundle sheath. So what they're showing is that in the mesophyll cell, that's where the plant will bring its carbon dioxide that it's taking in through the stomata, and it's going to react it with this molecule called PEP, P-E-P. I believe I have that written down in a future slide. So I will define that in a future slide. And so PEP is 3 carbons. Then you add a CO2 to that. Now you're up to 4 carbons. So that's why they call it a 4-carbon, a C4 cycle, because it's 4 carbons, okay? So what happens in the very first step of the C4 cycle is that you have PEP and CO2 reacting with each other. Now, Rubisco's not involved here. This is not something Rubisco can do. Rubisco cannot recognize PEP. So we have a different enzyme. It's PEP carboxylase. PEP carboxylase. I do need you to know what that enzyme is. So again, remember, ASE on the end of a word is an enzyme in the science class. And then you see the product of that is oxaloacetate, or you can also say oxaloacetic acid. Anything with ATE on the end of the word means it's an acid. So oxaloacetic acid. Okay, so let's go. Whoops, not there. Why did it do that? That is so weird. There it is. Okay. So PEP is phosphoenol pyruvate. Probably if I ask you something in class or on quizzes or exams, it's probably going to be presented to you as PEP. So it's okay if you write it that way. If you need to abbreviate something in an essay question down the line, it's okay to use abbreviations. Otherwise, your hand's going to get tired with some of these long words. So PEP and CO2 combine with the aid of the enzyme PEP carboxylase. So you can write it that way. So PEP carboxylase is really nice because this enzyme is very efficient. It only recognizes PEP and CO2. That's all it cares about. It does not care at all about oxygen. We say it has a very high binding affinity. That's the fancy way to phrase it. Very high binding affinity. That's good because that means as long as PEP carboxylase has some PEP and CO2 around it, this is going to happen. You're going to get the product OAA, oxaloacetic acid. Okay, so then what's going to happen is that you'll get oxaloacetic acid, OAA, converted to malate or malic acid. And then malic acid is going to go into the bundle sheath cell. So it's going to physically move. So this is from your book, this picture, and they're showing the, they're not even showing, well, I guess they are. They have the oxaloacetic acid, OAA, going to malate or malic acid, and then they're showing that it moves over into the bundle sheath. All right, the bundle sheath is down here. So I like to think of malate as a vector. And if you've taken classes where they've introduced that term to you, it usually means it's something being used to move or deliver something. And so you may have heard that malaria, a mosquito is a vector for malaria, the malaria pathogen or malaria disease. The mosquito moves from person to person and will transmit the pathogen for malaria. So think of malate like that mosquito, but instead of transporting and transferring disease, it's bringing carbon dioxide as part of its structure over to Rubisco in the bundle sheath. So it's a very important job. It's a vector. It's going to move out of the mesophyll into the bundle sheath. It has CO2 as part of its chemical structure, and it's going to be very nice and deliver that to Rubisco. And so that's what we talk about when we say it's concentrating CO2 around Rubisco. Malate's very important for that because malate's delivering that CO2 to Rubisco over and over and over. And that's what's causing Rubisco to be just swimming around in a bunch of bubbly CO2. So that means the PCO or photorespiration, the PCO cycle, is going to be suppressed quite a bit. We definitely see that plants that are C4, man, they're good at high temperatures compared to just straight C3. They definitely have an edge. However, if you take a C4 plant and you put them up here in the summer and grow them up here, they can't turn off the C4 pathway. It's a little bit extra energy and resources. So if you put a C4 plant and a C3 plant head to head in this type of environment, the C4 plants are at a disadvantage. So what I'm trying to point out here is the C4 pathway is an awesome adaptation for tropical areas, sure. But you take them out of their element, what they've been, their native plants are adapted to hot, dry, prone areas. Take them out of that, yeah, then this is going to be a little extra cost. You do see there's ATP that's needed for part of this. We'll get to that. They're using up some NADPH as well. So when they do the calculation, basically they say net 2 more ATP is needed compared to what we see with just the C3 cycle. So there is an energy cost, there's a resource cost, but if you are a plant and you are in tropical, subtropical environments, it's worth it. There is a payoff. The benefit outweigh the costs. So let's go back. For some reason, my brain likes this one better, so I'm going to teach off this one. Okay. So here is malate. It has CO2 as part of its structure. It's moving into the bundle sheath, and then it cleaves CO2 off of it and is very nice and gives it to Rubisco. Right, so that's where Rubisco is, right where the Calvin cycle is being drawn here. So did the artist put the Calvin cycle anywhere in the mesophyll cell? Do you see that anywhere? No, that's deliberate. Okay, so this is another thing that's spatial. C4 plants will run the Calvin cycle in the bundle sheath cell. Those cells will get the Calvin cycle cooking along, but out in the mesophyll cell, there's really not much of the Calvin cycle going on at all. So they're spatially segregating and kind of hiding away the Calvin cycle in the bundle sheath. The other key thing to think about, they also spatially separate the light reactions. So in the bundle sheath cell, they don't really do the light reactions. They just kind of devote the bundle sheath. I'm just gonna do the Calvin cycle here. And in the mesophyll cell, that's where I'll do the light reactions. So they're spatially separating where they do the Calvin cycle, where they do the light reactions, and where the kickoff to the C4 cycle happens. So as you can see, if Rubisco is tucked away in the bundle sheath cell in the inner part of the leaf, all these spongy mesophyll cells and the palisade cells, they're busy taking CO2 and getting it into the active site of PEP carboxylase. So it works out really well.
Okay, so mesophyll is where it kicks off.
Yes.
And the bundle sheath cell is where the Calvin cycle happens, and then the light cycle, where does the light cycle happen?
In the mesophyll cells.
Oh,
Yeah. So do you remember what is one of the products of the light reactions that I said, we don't really need to think too much about, but now we do? We enjoy the fruits of that product because it allowed us to exist, oxygen, right? So the light reactions are cooking along in the mesophyll cell, but not in the bundle sheath. The light reactions are producing oxygen. So oxygen is getting made in the mesophyll cell, but not in the bundle sheath. So you can see why the plants, it's like, that is great because now oxygen's not getting produced in the bundle sheath, which means Rubisco isn't being surrounded by oxygen. So they do a really nice job spatially separating some key things here. And when you look at all of that in total, basically what it's doing is creating a situation where Rubisco in the Calvin cycle here, the first step of the Calvin cycle, Rubisco is just getting fed a ton of carbon dioxide and there's no oxygen getting produced here in the bundle sheath because the light reactions aren't happening. Does that make sense? Yes.
AMP.
Adenosine monophosphate. So ATP means it has 3 phosphates, triphosphate. AMP means it dropped down to just 1. Usually you see a conversion of ATP to ADP. Yeah. So this is telling you, this is a really, I'm not going to get into it really, but just know this is a really funky enzyme that does this stuff. It's pretty amazing. So, all right. So, all right, so the C4 cycle is a cycle. So we're not quite done. Let me just finish the cycle here. So if malate moves over to the bundle sheath and gives off CO2 to Rubisco, there's another product that we need to think about after that, which is pyruvate. And so pyruvate is almost phosphoenolpyruvate, but not quite. It needs some phosphates attached to it. And so that's what this last step is to take pyruvate, it gets moved back into the mesophyll cell and this really funky enzyme works with it. And you end up with, the enzyme will attach phosphates to pyruvate and you'll get phosphoenolpyruvate. And then the cycle's back to the beginning. I don't know if I have I don't think I put it in here. Okay, so let me go back to that one. So once the phosphoenolpyruvate gets made, then it can all happen again. CO2 comes into the active site of PEP carboxylase, and then PEP comes in and they react and you get OAA, which gets converted to malate. Malate moves, delivers the CO2 to Rubisco in the bundle sheath. Pyruvate goes back and it just keeps happening all day long, as long as the stomata are open and CO2 is coming in. But these plants, what they're capable of doing, if you're a plant in the tropics and you're like, oh my gosh, it's getting hot, I'm going to close down my stomata. I'm going to close them down to like 10%. They're only 10% open. And so the rate of diffusion of CO2 coming in is going to be slower. It's just going to be slower. It's a smaller opening, more frictional forces. So CO2 comes in slower. But the C4 plants are like, I'm fine. I'm super efficient. I can do this. All right. And so as they're in the jungles and it's getting hotter and hotter through the day, they have this great ability to almost shut down their stomata, but they can keep it a little bit open and they can still keep doing this. And they're just so darn efficient. that it's okay. It's okay. So C4 plants, this is an amazing adaptation. And that's why C4 plants, when you look at where are they native to, it's usually tropics, subtropical areas where you can imagine the environment, is frequently hot, sometimes drought prone. And if you're on the equator, you don't have winter, so it's like, it's gonna be a lot of hot days through your life. Questions? Okay, so that's the C4 cycle. I'm gonna talk a little bit more about that next Monday, and then I'm gonna talk about the CAM pathway. But let's stop and think about the exam. So with the exam material, remember it's 1, 3, 4, 11 and parts of 14, right? Did I remember that? Oh, I'm good. So where are we having questions? Let me just say a few things as you're gathering your thoughts. So right now, the multiple choice is sitting at 40 questions. It might go up by 1 question, might go to 41, I don't know. Essay, there's 3 questions. 1 is just fill in the blank. And so it's not really essay, but I put it over on that version, on that portion. And then the other 2 are short answer. No big essay, but short answers. All right, where are we needing to get some clarification? Yes.
And like some of, obviously there's some overlap between the book and the.
Yeah.
What should we do?
In the readings?
Yeah.
So the readings. So chapter 1, I didn't do any PowerPoints. So, but that chapter is really short. There's only a few bullet terms in there, I think. So you're only gonna have a couple questions on that. But when you're reading, the way I would advise studying when you're going over the textbook reading is to pull up your PowerPoints for that chapter and start, reading through the chapter and kind of comparing. Because unless I say otherwise, the chapters are there for backup to the PowerPoints. The PowerPoint slides that I spend time with you going over, that's the most important stuff for sure. And then the textbook readings, it's a good textbook. And sometimes they present things slightly differently. So if it didn't click with the way I said it, maybe the book can be a little extra help. But unless I say you must know these terms and these pages that I didn't talk about, then I won't ask about those, if that makes sense. So continue to ask about that as we go through the semester. There may be some times where I'm in lecture and I'll say, oh, make sure to study this. I can't get through that section, but I need you to study it in the textbook. It just depends on how far behind I get in our schedule. So there's quite a bit of questions on the cells and tissues idea.
So I was going over like complex tissues and simple tissues.
Yeah.
And I know that when there's not tissues, like that, grass plants and watermelon plants don't have the vascular cambium, the cortices, but they have the inner calorie. But they still have xylem, right?
Yep. Sure do.
Yeah, okay.
It's different arrangements of where you put some of the there. We'll get more into that as we get into especially stem tissue. I'm gonna really dive into stem tissue. See some differences like that. Cells and tissues. Are there other questions on cells and tissues? A lot of its definitions. Remember to know which are living and dead, what makes it living versus dead.
Did you ever see if there's that tissue on like this in C2 elements or?
Well, know that they're so, no. I don't ask a specific question about design in the system.
Stacking?
Yeah,
Okay.
yeah, stacking, it's an end-to-end stacking that forms that conduit of cells through the vascular tissue and into the veins of the leaf. What about plant growth? A lot of terms in there too, plus like, okay, what does that do for the plant? You know, so it's not, it's terms plus. Like, what's the action that's causing that, or what does it do for the plant? Questions?
I don't necessarily have a question regarding to those, the more with the turgor movement.
Yeah.
Those are gonna be like on there as well. Are we gonna go? Are they gonna go like super in depth in like the dilo or motor prism or whatever it is, like the turf growth and like this?
Yes, so all those terms. We went over pigmotrophism. Isomonasty, pigmomorphogenesis, you'll know those terms and what they mean. And then I will be asking about how plants move. And so we know that there's more than one way. Turgor pressure is one way. What's the other way that plants can move? Maybe not super excitingly, but they figure out. So you have to sit there for a while to watch that movement, but it'll happen. It's a good way to spend some time if you just want to meditate. So turgor movements or growth movements. By the way, when we say growth, I don't know if I formally talked about this. When you say grow, it can be cell division, but it can also be cell elongation. Plants are good at that too. They can just elongate their cells and get physically bigger. It's usually a permanent thing, you know, I know. So they can do both. They can divide and then they can say, let's just grow these cells a little bit bigger. And then they stay that way.
How I feel in the winter.
Yes, exactly. So what about phytochrome? What about phytochrome? There's gonna be several questions on phytochrome. Warning, warning. So,
Mixing those up.
yep, being able to understand the
Yeah.
conversion back and forth, what does that do for the plant? Yes,
I'm just looking over my notes on PowerPoint, with like the sclerids, why are they called like stone cells again?
I don't know.
Okay,
I
I didn't know
think-
if there was like a,
In my, they should be called like grit cells, you know, like, cause
Like just a little bed.
they're just little, yeah, but it's more like a pebble or a piece of grit or, I think.
okay, there's not like a,
No, it's an old term,
okay, cool.
so. I tend not to use that term. I don't know if Dr. Robertus uses that term or not when he teaches, but.
Cause these old.
No, don't tell him I said that. He's awesome. All right, so what else? Let me go into. Do we have questions on phytochrome? Where's my plant growth? Plant growth. Do we have any questions on phytochrome? Yes.
Can you just like go over actually, so again?
So, what do you see phytochrome doing on here? What's it doing? It's changing its shape, right? So that is a key concept. It's pretty elegant when you think about it. Just changing its shape makes all the difference in how the plant perceives what's happening. And so you go from the M shape, where that's the so-called phytochrome form, that's PR. So P stands for the phytochrome photoreceptor, R is the red version. But what that means is it's all set up and designed to maximally absorb red light. And then when it does that, it shifts over to the Y form. So PFR is the Y form. That's the phytochrome receptor that's just really good at absorbing far red light. And if it does that, it's going to physically shift back to the M form. And so you have this toggling back and forth just based on what quality of light is there. And so it really is, the plant is constantly assessing the ratio of those two, like how much PFR phytochrome do I have? How much PR form do I have? Based on that, it's gonna hopefully tell the plant to behave a certain way to respond appropriately to whatever's going on around it. And so here you're seeing the PFR form is the so-called active one. That means it's actually shifting and interacting with proteins in the nucleus for gene expression. Yeah, think through that, the flower, no flower, think through that experiment. In the PowerPoint slides, we had ideas going on with that of how the plant is dealing with getting hit with yes,
Well, anything about propagation?
yes, a little bit on propagation. By the way, I went over, I couldn't see the actual hard stats on the lab quiz you all did just finished today. But it looks like you did really well on the stuff that's multiple choice or drag and drop. I don't know about the essay yet. But I noticed there were some propagation questions on there. So it's like those propagation questions on the lab quiz would help you on Friday. There's a couple other questions on cells and tissues that, but yeah, that's good review.
Are you gonna give us like pictures like on the lab quiz?
No,
Okay.
not like that. That's saved for the lab.
Okay.
Yeah.
Okay.
So it's not-
Because I-
Identify?
I was
No.
A little nervous.
No, that's the lab portion.
Okay.
Yeah.
All right, so I will be checking my email into the evening hours from now until, well, and Friday morning, of course. If you have a question that you want to send out to me via email, if you're at home at night, send it out and I'll try to respond as quickly as I can. Okay? But just keep studying. Come prepared.
Also, if you, whatever, if you plan on making